Welcome to this session of the USC AI Future Symposium on AI and Data Science. It is my pleasure today to introduce Regina Barzilay, who's going to give a keynote in this session. Regina is a School of Engineering Distinguished Professor of AI and Health in the Department of Electrical Engineering and Computer Science at the Massachusetts Institute of Technology. She's also a member of the Computer Science and Artificial Intelligence Laboratory. Regina has received many awards, including an MIT Technology Review TR35 award. She's also received a MacArthur Fellowship, and last year she was the inaugural recipient of the AAAI Squirrel AI Award for Artificial Intelligence for the Benefit of Humanity. Regina received her PhD in Computer Science from Columbia University. She is a fellow of the Association for Computational Linguistics and a fellow of the AAAI. She's going to tell us today about her work on AI for drug design and other adventures in the biomedical area. Regina, thank you for being here.
Thank you very much, Yolanda, for inviting me over and for all of you taking the time to listen to me. So today I will introduce you to a relatively new but very fast developing area of AI for drug design. And I will talk to you where the field is now and what are the open questions in the field and what we can expect realistically this field can deliver. So I know that many of you are not very closely familiar with this field, but actually it's very interesting that when you are trying to understand how the drug design is done today, you can understand why there are so many diseases for which we don't have good treatments today. So the space of potential molecules, like even small molecules that you can use for treating the diseases is 10 in the 60s. It's humongous space. Of course, you cannot directly go and explore them all. And what typically happens is that there is some human intuition that drives this discovery process. So for instance, if you're a medicinal chemist, you may look at the molecular graph and say, you know, this particular subgroup drives this activity. So here you can see an example of a representative of antibiotic family. And here you can see this particular subgroup that often appears in antibiotics because it has antimicrobial property. On the other hand, if you're a biologist, you can actually talk about the proteins that are involved in the disease, and you can talk about the binding, how you can modify the function of this protein by finding a small molecule that binds to it. But whatever you do, however you do it, it's really not precise science. There are a lot of uncertainty in this kind of descriptors. And what people typically do, they supplement this intuition or this knowledge with a lot of experimentation. And there is a lot of screening and experimental work that always involved in drug discovery. Now, you can say that, of course, there is a lot of progress in the experimental volume, but even with the most involved robotics that you can today have in pharmaceutical company, you can maybe screen, libraries of millions of molecules, but you clearly cannot empirically explore the space of 10 in the 60s. It's like just too much. Of course, this is a place where AI and machine learning can really make a difference, because the question is, can you actually predict what this molecule is doing without manufacturing it, just doing it in silico screening of the big space to identify a small subsets that need to be tested experimentally. And I should say that I started working in this field in 2015, and here you can see the graph on at least like two very prominent machine learning conferences. In 2015, there was no papers on modeling molecular structures, but you can see over the years, it gradually continues to grow, and now it's even a higher number of papers. It is really a sub-community in AI that works on drug discovery and modeling of molecular structures. And one of the key capacities that this method delivered today is something which is called property prediction. And by property, I can mean anything ranging like from solubility, like physical-chemical property, to some specific bioactivity, like killing cancer cells. So what you want to do, you want to train the model to predict this property so that you can perform the virtual screening, like I described to you, before you select molecules which to check experimentally. But you may also want to do de novo design. And when you just say, I want to have to generate from scratch a molecule with certain property, and here you will see in the second part of my talk that, you know, having access to property predictor really makes a huge difference. So let's just think about it from AI perspective. I am a computer scientist. I'm not a chemist or biologist. So typically what comes to my hand after the experiments were done is a selection of like 2D structures of molecule and some numbers. So let's say in this particular case, this number may capture antibacterial activity of this molecule. And what are you trying to do? You're trying to learn the mapping from the structure to this particular activity. And you can ask, is it like really new idea? And I would be the first one to say, absolutely no. The idea that you can go from structure to activity is quite old, it's decades old. But the way people did it in computational chemistry is actually quite distinct from what we are doing today. So the idea there was to take the molecule, be 2D or 3D graph, and then you would summarize it by hand into a vector. And this vector would record different substructures that are present in this molecule. And you can say, how do I know which substructures to use? You can say, because you are an expert, you know something about chemistry, you would identify this type of substructures, and then you can apply any machine learning methods to combine them together and predict the property. And the main change that came is actually say, you know what, as a human, we have very limited understanding of what are the good substructures. We don't know. So maybe the key step here is to develop a machine learning method that can take molecular graph and then automatically learn how to, to summarize it into a vector from which you can do the optimal prediction. And in this particular case, if you take the same molecule, and in one case you're training that for like toxicity property, and in another case for solubility, you may summarize it in a different way. And again, what you're losing here is interpretability, because you don't really know what, the first coordinate means or what the second coordinate means. But what you're hopefully gaining is the ability to predict much better the desired properties, which goes beyond human intuition. And if I'm to draw it pictorially, I would say we're actually learning to translate the molecule, to embed them in some high dimensional space where structures, molecular structures that have similar property, their vectors live together, and for those which have different property, they live farther apart. We hope that we can build this kind of translation. Or in other words, we hope that we can learn the embedding of this molecule in this high dimensional space, such that there is a linear relation between embedding distance of molecule and the property values, and distance in their property values. So that's what we hope to get. And, there are many algorithms to do it. I would illustrate you one that for those of you who are familiar with graph convolutions, it would look familiar. For those who don't, it's also very intuitive. So again, what I'm trying to do is translate my graph into a vector. So the way I will do it, I will start with every atom having its own vector. And here I am, as a domain expert, will decide what features are of the vector. This is a place where, as a domain expert, I can say something. And the features that I can use would be very simple: the type of the atom, the degree, and so on. So now every atom is a vector. It only knows about itself. It doesn't really know about the neighborhood. And then the second step, what you would do, this is a second graph convolution step, you would actually try to combine information from the surrounding neighbors. So what I can do, I can have my own vector, let's say I'm vector V, the red one, and then my neighbors has their own vectors. And I would learn how to combine them together into a single new vector for me. where I incorporate information from my neighbors. And you can say there are many ways how you can do this combination, but of course, in this case, I will combine it in a way which is guaranteed to create a representation which is optimizes my prediction property on the training set. And of course, it can be done properly in the algorithmic terms. So in summary, what will happen after one step, I am gonna not only remember information about me, but in some ways information from my neighbors. We're gonna do it for all the atoms simultaneously. So now everybody know information about one hop neighborhood, and then you can continue this process. And now, since everybody are updated, you're going to have information from 2 hop neighborhoods seeping in, and so on and so forth. And eventually, you know, every atom is going to have this kind of updated vector. You're going to sum them up and get a vector for molecule. So this is like one kind of cartoonish description of how you can translate graphs into these vectors. And, you know, people who know chemistry or biology, they're in the audience, they always ask this question, you know, but we really know that molecules, they are not 2D. That's how we draw them in our chemistry, you know, high school chemistry, but they're actually 3D. How are we dealing with it? And some of the 3D features, like stereochemistry, you can explicitly incorporate in the features of the atom, but I will come back later about trying actually to model the whole structure in 3D. And I should say that we, one of the models that we've built here, you can see the publication is very extensively used today for virtual screening in pharmaceutical industry and in research. We very extensively tested it, you know, at least at the time of the publication. I think now, you know, it performs reasonably well. But of course, you know, as we know in AI, you know, the models, state of the art are constantly moving, but this is kind of a very reasonable starting point for many explorations. And I can tell you that besides writing a publication, we also used it to identify an antibiotic drug. It's called halicin. It has its own Wikipedia page. And this particular drug has very strong antibacterial activity against multiple pathogens, drug resistance pathogens, which are identified as critical by World Health Organization. And what was interesting about this drug, that we actually discovered it in a very limited budget. Our training set contained of 2,500 molecules, which were screened against a pathogen called E. coli. And for those of you who've never seen screening, you can see here this kind of gray with this bin thing. It's actually the screening results, where each bin corresponds to different compound, which is placed to interact with this E. coli, and depending whether it killed it or not, you would have a different color. But at any rate, remember what I showed you, we would have every molecule and a number that tells us how much this compound kills or doesn't kill E. coli. And the picture on the very bottom is a postdoc, the first author of the study, Jonathan Stokes, who actually did all the experimental work. And we were very lucky here that the compound that we found actually was active not only against E. coli, but it's also work against C. difficile and A. baumannii, which are really nasty kind of bacteria to have. And many existing treatments are not very effective against them. And the reason we believe that this halicin is actually doing such a great job killing this pathogen is because it has very different mechanism of killing the traditional antibiotics. And I should say it was not necessarily, you know, the machine learning brilliance, but it was more the brilliance of Jonathan Stokes that created a training set that not only had antibiotics and everything else, but he actually took a variety of compounds like natural products or cancer drug or other things, so that they have different degrees of antibacterial activity. So the model can really learn from a wide range of chemistry what may result in killing activity. And he selected from the set of all the prediction compounds that don't look at all like current antibiotics. And that's what created this very interesting mechanism of action. So now I should say that, you know, the wrong conclusion from what I described, that the problem of task of, you know, this virtual screening and property prediction is solved. It's very far from solved. And I want just to illustrate some type of questions, which are really open-ended questions for which we still don't know the answers, and show what progress are we making. So one big question, which I was actually not ever thinking before I started working in this area, is a question of confidence estimate, because each molecule that you want to test, it actually takes time and money to synthesize. If the model tells you, this model has very strong killing capacity, you actually want to know what is the confidence of this model. And in traditional medicinal chemistry, there are a lot of work on various types of confidence estimate methods, like mean variance estimation, or you can just look at the embedding distance between your training set and testing set, variety of them. There are lots and lots of literature about it. So when we try these techniques for our deep learning models, what we discover that these techniques really don't work to the tremendous extent. Like, for instance, we found data sets that if you select top, let's say 10% of most confident molecules and test on them, you actually get lower accuracy if you are directly using the simple threshold. So this is a big area, how you can predict to do reliable confidence estimation. This is a paper below that describes our current, these findings, and we are working very hard at MIT on conformal predictions, which can actually give you guarantees. And there is, a lot of need to continue put, technical effort into this area. So another important question is how you can do low resource training. And the reason you need to do low resource training, because, whenever you are moving to a new therapeutic area, you may not have enough training data, and you really want to be able to predict well. And even if you look at our training data, it was just 2,500 compounds. It's a tiny training set. And we know that there is one area where we learned a lot how to deal with relatively small amount of data in natural language processing, and in computer vision is really using pre-training. For those of you who are not familiar, the idea is kind of very intuitive. You can say, I'm going to have an auxiliary task where I have a sentence, I remove, I mask some words, I try to predict them. And because I have lots and lots of sentences in English, I can create humongous training set. And when I'm training my deep learning model to do so, I can actually learn very good representation for words. So in such a way, again, that the words with similar meanings, their vectors would be close to each other, intuitively speaking. And there are lots of results. This is not even the most recent results, that if you're looking at the whole bunch of data sets, if you compare how well do you do if you are using or not using pre-training, the difference is really tremendous. You can see, for instance, that in this whole bunch of 11 different tasks, whenever you are adding pre-training results, you actually outperform human performance. You can say, wonderful, I can now take these ideas of masking, apply it to chemistry, and see what it does. And there are also humongous data set of this kind of molecular structures. It's called zinc. I think it has like hundreds of millions of compounds. And there was a work from Stanford that tried to really replicate the same idea of masking to this data set, where you're removing an atom or removing a functional group to try to predict it back. And the results are very not satisfactory. It really doesn't show significant improvement that we see in natural language processing and computer vision. And I still myself don't know, how to make it work. And despite the fact that it's an open problem in the field, we still don't have a technical solution. But I want to show you that some ideas that kind of, inspired by kind of development in the core machine learning field are really deliver very nice results when applied to chemistry. For instance, generalization is really a big deal whenever you are dealing with, discovery. Why? Because typically, you have your screening data in one part of the chemical space, but you really want to apply it to another part of chemical space. And for this reason, again, in contrast to NLP or computer vision, when you decide about training, development, and testing, you don't just split them randomly as we typically do. We split them based on scaffolds. Scaffolds are kind of like the core part of the molecule. So you want to make sure that what you're training on doesn't look the same as what you're testing on. So it makes generalization very hard. And this is an area where ideas from invariant learning invariant representation really played very nicely. So here you can say, I'm just gonna emulate during the training the same scenario where I divided my training, let's say, into two domains which look distinct. And I try to learn representation in such a way that I can do well for the scaffolds that I haven't seen. So this idea works very nicely, and an area where I expect to see, more work coming here. I just put a reference to our paper. And again, there is a lot of place to really improve here. But one area that I was sort of forced to get into, and I think the whole community got forced to get into, is how to learn representation. And maybe a year ago, if I would have given this talk, I would start talking to you, instead of using graph convolution, I would show you some alternative representation and talk about it. But, what I realized in the last year, that we really need to learn representation and to understand what's going on in biological terms. Because let's go back to the representation, how I told you the story. The way I told you the story, I told you, you have this, graphs, and you have a number. I don't care how the number come about. I just tell you that you have it and you need to learn the mapping. But what we know that actually this number doesn't come from nowhere. There is some biological mechanism. For instance, maybe if it is bioactivity, there is a protein, your molecule does binding. There is a lot of understanding about it, but we are totally throwing it out by just summarizing it by one number. And the reason we sort of were really forced to look at it more closely when we were working on COVID antivirals. So it is quite well known that for many viral diseases, combinations are really working very well, because they can kind of simultaneously look at multiple targets which are relevant to the virus. And, and since there was no single compound that did wonders for COVID, we thought, why wouldn't we design combination? And we started collaboration with NCADS, which is one of the institutes of NIH that looks on the, drug development. They provided us I think less than 200 combinations, which is very little, correct? And the question is how you can learn when you have so very few combinations. We had a lot of data for individual compounds, but we didn't have enough combination data. And the way people, how people define what is a combination, synergetic combination, you kind of try, and there is one example to do this called Bliss formula. Bliss doesn't mean that somebody is blessed. It just means the name of the person, but pretty much you are trying to find combination of drugs that do more than the sum of the two parts. And there is a mechanism to directly kind of compute it. And what we wanted to do is to look at two molecules and say, would they have a synergetic interaction? And it's very hard to do directly if you just have 200 points for training. So what we did do, we actually built on the knowledge that was available at the time, and it's still available, and done by people in biology, which instead of just looking at the number kill or doesn't kill the virus, had a lot of information about relevant targets. They understood how the virus replicates, which host proteins are relevant, and so on. And we thought that since the synergy is all about operating on different targets, we can make our model not only directly go from the structure to the antiviral activity, but actually force the model to tell us which biological targets are involved. So you can see that what you're doing, you're starting with your structure, you're learning the representation that tries to predict which targets are involved, and then from there you are predicting the activity for each single compound. Now, the truth is, for those, again, who knows biology, that information about targets, it's quite noisy. We don't know all the targets. So instead of just, having this list of possible targets, we're going to have these latent targets. In other words, we are summarizing our molecule into a set which explicitly indicates targets that we know about, and also some set of hidden variables. And it gives more expressive power to the model. So in summary, what the model does, it will translate each compound into, when modeling its activity into this kind of biochemical latent representation. This is for compound A, for compound B. Then you concatenate these compounds and you predict the activity of the combination, and then you can directly optimize Bliss score. Because the data for target activation and for activity of a single compound is available and plentiful, you can still learn synergetic model even though you have few hundred combinations. And I should say that the institute and CATS with which we collaborated was particularly interested in remdesivir, and we found a number of molecules which really boost the activity of remdesivir. And you can see that, for instance, for remdesivir and IQ1S, each one of them was not fully killing the virus, actually very partial, but when you combine them together, they had very strong killing activity, and our NCATS experiments confirmed these combinations. So this was actually a very nice result that shows you using biology helps. The interesting parts of there are a lot of things going on in this graph, but I just want to bring to your attention one point, which is like this middle thing with the brown and the, I don't know, violet color, that actually shows that you can do this model without explicitly assuming that all the targets are latent, without incorporating these biological models. You still get some results, but you definitely benefit from adding to the model what we already experimentally know is true. And this is, for me, was really an eye-opening moment, that we have to incorporate information that we know is, is out there. And there are a lot of interesting questions on how to do modeling. We know already that, there is something which is called property cliff, when you just change one atom and all of a sudden, the property really goes down or up. So it's very non-smooth transition. Why does it happen? Because molecules actually have 3D conformers, and changing what it looks to you like one small change in 2D may result in very different configurations. So we work hard on modeling the 3D conformers and making 3D reasoning. There is also, a big question of how do you model, these proteins to which the molecule binds. In the previous presentation, what I showed you, we actually said target 1, target 2, target 3. It's kind of one hot embedding. You don't really understand, what is the relation between target 1 and target 2. You just model them as separate entities. Of course, they play a role, and the question is how to incorporate them. And this question was, was of great interest of mine. And the reason is that there was this papers published at Harvard and Stanford and other places. Maybe some of you who are interested in this, line of work seen it. It's called about, network medicine framework for drug repurposing. So you can imagine to yourself constructing a huge graph where you can have, all these targets, all these proteins, the relation between the proteins and disease, which proteins are affected by disease. You also know which small molecules affecting each target, each protein. And you have this very big thing. You can put all the known targets, all the known molecules, like known drug molecules, all the diseases. And then you have these big graphs, and you can try to do inference on this graph, like for instance, with graph convolutions, to identify, compounds that are likely to have desired behavior. I was actually very puzzled, because in this case, you're not modeling molecular structure, you're not modeling protein structure, they all are kind of one hot embedding. And I was really surprised, how come does it work so wonderful when you are representing it in such a simplistic way? So, and my student Rachel was also interested, kind of intrigued by this question. And we took something which is called DepMap. This is a very large collection released by Broad Institute related to cancer, where you have all the relevant targets and you have the compounds and the different types of cancer. And she did this study. And actually what she discovered, I would not go in the greatest detail, but what she sort of discovered, that the reason it works so well is not because, there is some magic in the graph convolutions, but the main reason is that you are pretty much memorizing the targets from the training and you are directly utilizing to do testing prediction. So you don't have really capacity to generalize to new settings, as you can see here. But if you are operating within the same domain that you've seen in training, you can actually do something. So it was very unsatisfactory in terms of empirical results, but it really shows you a very strong motivation, how important it is to properly model these targets, to properly model these proteins. And here I have a sequence of slides that actually demonstrates that, you know, that the question of how do you model this iteration between the protein and the compounds really matters. And this is truly a computer science AI question, because of course we know that proteins are 3D, but you don't know the 3D conformer. So the question is how far you can go with 1D. And you can check it empirically by looking at different kind of combination. Here you're given a small molecule and given a protein, and you try to see whether they bind or not. And you already know, I described to you, you can take the molecule, you can summarize it with GNN into a vector. The question is, what can you do with a protein? So the first thing that we can do, we can just kind of code proteins based on their identity, 1, 2, 3, 4, 5, you know, with one code encoding, and train the model. You cannot generalize to new proteins, but you can hopefully do it with proteins that you've seen in the past. The next thing that you can do is can say, okay, I know I have a sequence, it's 1D sequence. I can just use my, you know, language models to represent it, to compress it into a vector and then make predictions. And there is a lot of work on doing this kind of encoding. And then finally, you can actually do more, and you can say, I switched one slide, and you can actually do more, and you can say, you know what, when I'm modeling this and doing this compression of the 1D sequence, I want to identify the site where the small molecule binds to a protein. It's called binding pocket. And this information is available during training. And you can use attention of the model to kind of to make sure that this is the site that you're looking at when you're making prediction. And what you will see on the numbers below, that of course, you know, the things that we expected will happen. The more elaborate you model in terms of capturing the protein structure and capturing the interaction, you know, the better is your performance. And this was just kind of a small exploration, but there is a lot of we can do again on the computational side to truly improve it. And the final thing that I want to say about the property prediction, it's not yet the end of my talk. The final thing that I want to say about the property prediction, that what I discovered when we worked on halicin, which was very surprising to me, that, you know, we saw already this molecule kills, they put it into the mice, everything was great. But then they need to explain the mechanism of action. You know, how exactly does it kill the bacteria? And it took to the postdoc, to Jonathan Stokes, you know, a good 4 or 5 months to do it, because it's really exploratory process. So in words of machine learning, what you're trying to do, you are trying to provide a rationale of your prediction. Why do you say that this molecule killed this pathogen? And, you know, and there are ways to characterize this mechanism. So now we develop models that can actually not only tell you this molecule is going to kill the pathogen, but it can also tell you what kind of features of the mechanisms are there that enable this killing. So then when somebody wants to really investigate the mechanism, you're very much limiting the space of possibilities that they need to check. But in my final few minutes, I want to talk to you about de novo molecular design. And, you know, again, we all are using Google Translate, and we are using a lot of generative models. You know, you can think to yourself, you know, we take a sentence in English, it's summarized into a vector, and then from this vector, we generate a sentence in Spanish, correct? So this is a generative kind of model that we all use. Of course, there is a lot of space to apply this kind of generative model in the molecular space. Why? Because even if you have like all the machines that you can get, you know, screening virtually 10 in the 60s is not a good thing. And typically you're not optimizing for one property, you're optimizing for very many properties at the same time. Like you want to make sure that it's active, that it's soluble, that it's not toxic, blah, blah, blah. So it's a lot of properties that you care about at the same time. So, I want to show you, we have many models that we developed at MIT, but I want to show you one model which I find kind of particularly interesting because it in some ways mimics what medicinal chemists do when they generate new molecules. And this model is particularly applicable again when you try to do multi-parameter optimization. When you have many different kind of items on your agenda, you have a very big desiderata of what you want from your molecule. So the way humans do it, you can kind of start, you do it like a Lego construction. You start with whatever you start and say, okay, I'm going to add this piece to the molecule because it's going to achieve this property. And then I'm going to add this other piece because it's going to achieve the other property. So you kind of combine them together. You're making sure that they don't contradict each other. And you may add some other atoms to create kind of a coherent structural molecule to create a filler. So what we would want our model to do is kind of emulate this type of behavior by answering two questions. The question number one that you need to answer is, what are the pieces that kind of drive certain properties that I want to have? You know, like let's say I want to have this killing of this bug activity. Which type of subgraphs of functional groups drive this property? So you would have maybe an kind of a bag of pieces, of Lego pieces that correspond to the property. And then the second question that you need to ask is, okay, fine, I have it for property one bag, I have a bag for property 2, for property 10. How do I take them together and generate a nice coherent molecule? And note that you may never see in your data even one molecule which have all these desired properties. You would have a screening data for the first property, for the second, for the 10th, but not something which have all the 10 properties. So I will show you a method that can do it. So the first question that I told you about is kind of to create the bag of pieces for the first property. So what you're trying to do is, in computational terms again, is to find the minimal fragment, the rationale, that drives this biological activity. So in other words, what you want to do is to say, I want the smallest fragment so that if I have it, my molecule is still active and this piece is connected. In other words, if I'm now going to pick it out of the molecule, the property is going to drop. So this is a piece that I cannot eliminate from molecule without compromising the property. And, there are many ways to do, it's like rational in natural language processing. For those of you who are more familiar, when you're trying to say, what is a fragment of the document that makes this review, let's say, positive or negative? Again, many ways to do it. In our particular implementation, we use Monte Carlo tree search to do this identification, but again, lots of possibilities. But what you are going to get when you finish doing it, for each property, you are going to create this fragment vocabulary, many possible pieces that if you add them, they're going to drive your property. And then the next step is to select from each one of the bags, the pieces, so that together, whatever you get is going to be, a nice molecule. And it's like a sentence. You cannot just stack them together. You need to have some kind of chemical rules to make sure that this is a reasonable molecule. And in this case, we use hierarchical generation model that, is slightly different from the graph convolutional model that I described earlier, because you would have, different level encoding. We would have encoding of the individual atoms and encoding of the fragments. And the convolution now is going to have these two layers where the information is propagated. And eventually, your generation process would be, you would start with a piece, and then you would identify the point to which you are going to be adding the new piece and predict the type of piece that you're going to be adding. So it's a kind of piece by piece generation. So we're going to be growing molecule one fragment at a time. So this is the idea of this hierarchical generation. I want to say that this particular mechanism, whenever we're doing this multi-parameter optimization, is currently achieving state-of-the-art in generation. And you can ask, what can I do with this type of model? For instance, what you can do with this type of model is to create narrow spectrum antibiotics. The reason nobody likes to eat antibiotics, because they're like a bomb. They kill the pathogen, but they kill everything else. So here you can say, I want to have a drug which kills one pathogen, but leaves everything else intact. All the good things in my body, it leaves intact. So you can formulate it all as properties and learn them. And in conclusion, I just want to say that the key collaborators in this work is Wingong Jin, my student Wingong Jin, which is co-advised by Professor Tomi Yakala. And the final slide, I want to say, big thanks to this consortium, which is machine learning for pharmaceutical discovery and synthesis. They not only, supported this research, but they actually are very big users of these tools. Almost all the tools that I presented you are currently used by these companies. And I learned a lot by talking to them and, getting their feedback in how these tools should be developed. So thank you very much.
Thank you, Regina. What an inspiring talk. Please, for the audience, please add your questions to the QA panel. And I'm going to kick off the discussion by asking you about explanation. So there's a lot of interest these days on creating mechanisms for AI systems to explain themselves, particularly machine learning systems. And you talked a lot about using prior knowledge, about understanding how we describe the mechanisms by the way how these molecules work. You didn't talk about providing explanations. It seems that as long as you highlight the features of the molecules that can be mechanistic, that might be sufficient. Could you talk a little bit more about how much you are pulled into the explanation world versus this works and that's sufficient?
Yeah, so I think this is actually a very good debate especially when we're talking about kind of precise sciences. And the good question is, what is the goal of the explanation? Many times, people, like I'm talking about scientists in chemistry, cannot write precisely certain rules, why some molecules are soluble and why others are not. They have some set of guidelines that can explain some of the phenomena, but not the whole phenomena. So it doesn't strike me that it's even feasible to write the rules that humans who spent decades thinking about it, that the model will discover that human didn't think. It's very intricate, interdependent, patterns that I'm not sure we're even going to benefit by getting them. And I personally always prefer to have models which have better generalizability, which have, higher accuracy than a simpler model, which can explain exactly what it was doing, but it's actually not doing great. But at the same time, there are certain mechanisms that are related to our understanding. Like you can use a system we all use, in Amazon or whatever system that like ranks the reviews or something like this, without understanding why it was ranked high or low. But for instance, every drug that is put on the market, they have to write the mechanism of action for the drug. So you can think, is it a rationale? Yeah, at least it's some form of rationale that should be for the, biologist or clinician provide some insight of what's going on. So I think it is, useful to train our models, not to only give prediction, but at least give some insight, which would be sufficient for that professional to do the empirical work that will truly elicit the key mechanism. But there are two parts of it. One is to explain to us, correct, what's going on. Then there is another part. If we already know something because we understand chemistry or we understand physics or we understand biology, there is absolutely no point not to inject this information into the models. And many times you still can, get good prediction even if you don't do that, but it just requires a lot of data and it reduces your ability to generalize. So what I discovered, especially through this year working on COVID, where the data was very limited and fragmented, the more data, the more mechanistically rich are your models, the better you can generalize.
Thank you so much. You've been working for many years in natural language processing, and there's always been efforts to generalize natural language processing techniques to biology, particularly genomics. It's an encoding in some ways. So I wonder how has all this experience with natural language positioned you differently to look at these problems of drug design and drug discovery, and how has working on this area of biology changed your approach or your thinking about natural language?
I actually think it was a very good opening point that when I started working in this field, that I did have, natural language processing view. When you see something as a sequence, or you see it as a tree, or you see it as a graph, because what happens in natural language, and I think in some ways it is sad. I mean, it resulted in great performance, but in some ways it kind of a bit sad and disappointing that, when I was a graduate student in junior faculty, there was a lot of model, there was a lot of emphasis on bringing syntax, on bringing semantics, and how to build a model that, incorporate what we human believe is a structure into the model. What we think happen now in natural language processing, that the top performing model actually do not explicitly utilize human representation of the structure. They're not based on the syntactic tree, as, linguists will think of it, though they do incorporate it. I mean, it's sort of captured indirectly by looking at the sequence. So, but when we started working on chemistry, we immediately start thinking, what is the right structure that you're going to capture a molecule? And the surprising difference from NLP, that actually incorporating the structural approaches does help. So talking about like molecular generation, you can just say, I have a vector and I'm just going to generate the full molecular graph. This approach actually doesn't work that great. You can do it, but it's very much suboptimal. On the other hand, if you are operating on these multi-layer approaches where you have a high-level structure which you further specify, which is sort of similar to some extent to the syntactic trees, it actually results in much more efficient generation. So in summary, maybe because there is not that huge amounts of data that we see in natural language processing, in chemistry, which is very rewarding, the smart modeling can compensate for the lack of data and result in better accuracy. So I very much enjoy it.
Very good. I'll ask you one last quick question that somebody sent. I always say that I hope AI will make us better people and help us improve how we work as humans and as a society. And they were asking that they were understanding that when you were searching for halicin, that one of your criteria is that it would be a drug that had no IP restrictions or could be made freely available. Is that right? And how dense is the space in drug discovery of things that are already proprietary or not?
So there is a huge space. There is really a huge space. Think about it. You have billions of molecules out there that each one of them can be a drug. So maybe the one thing to note, that the reason antibiotics are of such a great interest to AI techniques, that we as a society don't have many options despite increasing resistance, because they are economically not valuable for pharmaceutical industry. There is no good pipelines, and the beauty of it that whatever you can put and on the market with some non-profit, or make it open and economically viable, can really help. So I think that there is a huge space here to go into IP free space and identify candidates which one can put for very low price range into the market, enable lots and lots of people who are currently suffering from the lack of such treatments to utilize them.
Okay, I'm going to end the session here. We're out of time. Thank you so much, Regina, for a very, very inspiring talk. And we will resume at 15 minutes past the hour with our panel on AI and health, and we'll continue the discussion. Thank you.
Thank you very much, Yolanda. Thank you. And thank you all for listening. Bye.
Thank you, everyone. Bye.